The cannabis industry is rapidly growing, and its growth is outpacing old agricultural techniques. Historically, cloning is the method that has been used in large-scale cannabis production. Unfortunately, cloning can cause problems for some industrial crops, especially in the case of annual crops such as cannabis. Micropropagation is an alternate method of plant reproduction that avoids many of the issues of cloning, but it can be a costly and time-consuming process. Pharmagreen is pioneering a new version of this process called the Afreen method that reduces the cost of production and shortens the time between growing cycles.
That particular system takes traditional 4 stages of micropropagation in the tissue culture technology and combines 2 of the stages to make it a 3-stage process.
This proprietary technology makes micropropagation a more practical form of agriculture. Despite its drawbacks, cloning is currently the most common form of plant reproduction for cannabis. This process involves cutting stems off an adult mother plant, applying a rooting hormone, then planting each one in a medium that stimulates the plant into growing roots. Once rooted, the plant is then transferred to soil where it continues to grow until ready for harvest. Despite the simplicity of this process, numerous problems can occur.
Cloning process is relatively easy and simple, and it doesn't involve any complicated or expensive equipment. And if done correctly, usually we will get a root in 8 to 12 days. But the disadvantage associated with this method are quite a bit.
The most obvious shortcoming is the number of clones cannot exceed the mother's viable branches. For a smaller operation, this may not be an issue, but once cloning is taken to an industrial scale, it quickly becomes impractical. A less obvious issue arises with repeated cuttings to the mother plant. When cut, the plant releases hormones that can alter its genetics over time.
So as the more tips you cut to make more clones from it, they're not the same. Their genetic material starts to change, their hormone levels start to change, because now the plant is trying to excrete hormones for the survivability of the plant. By cutting its tips, it's basically the plant thinks it's dying.
These genetic changes can drastically alter the plant's qualities, such as its potency and the exact kinds of cannabinoids produced. Combined with naturally occurring genetic variation, a cloned strain selected for its qualities may have little resemblance to its descendants over time.
The reason is because they are taken from different mother plants, which itself had genetic variation. So when you take clones from those mother plants, grow them, variation rate is so high that it is completely at some point very difficult to use them.
Micropropagation is a solution to both these issues by reproducing the plant at a cellular level. Micropropagation begins in a sterilized lab with stem cells taken from the developing leaves of a selected plant. These samples are then placed in test tubes containing hormone gel. Over the course of 3 to 4 weeks, the cells begin to multiply. Once the cluster of cells is large enough, it is divided and transferred into larger jars containing a different hormone gel. This new hormone stimulates the cells into growing stems and leaves over another 3 to 4 weeks. At this point, the plants are removed and transferred to another jar containing a different hormone that causes them to develop roots. After the plantlets have matured, they are transplanted into containers of soil and left to acclimatize in greenhouses until they are ready to be sold to licensed producers.
Because for micropropagation, we need a very small amount of material, and we can, even if we take it once, that's enough. We can preserve it as a stock, and we can use it repeatedly for producing new starting materials.
One issue with micropropagation is the number of steps and resources required to produce these plantlets. The Afreen method used at Pharmagreen's biotech facility is able to combine some steps of this process, cutting costs and reducing time between growing cycles.
The facility that we're building, Pharmagreen Biotech Complex, and its advancements in agriculture technology that can not only be used for cannabis, but the whole agriculture sector as a whole. And the economics further importance to that is that this facility is a recession-proof facility. recession proof in the sense that today we're producing marijuana tissue cultured plantlets. Maybe 2 years, maybe 5 years, maybe never, but maybe we'll be producing tomatoes or saffron. So we can produce these different species of plants without having to change the technology.
With this streamlined agriculture system, PharmaGreen, in partnership with Alliance Growers, is on track to become leaders in the rapidly expanding global cannabis industry. The Afreen method, along with other proprietary technologies, will set a new industry standard for quality, consistency, and production. For more information, please visit alliancegrowers.com and pharmagreen.ca.